Hi, good morning and good afternoon. I will present a short slide deck about chromatographic tools for isolation of extracellular vesicles and process monitoring analytics. So how do you isolate your extracellular vesicles? We use monoliths. Via separations is the only via separations, now part of Sartorius Group, is the exclusive producer of CIM monolithic columns. CIM means Convective Interaction Media. We have over 22 years experiences and over 200 employees. We are specialists in the purification of large biological molecules such as AAV, adeno, influenza, pDNA, mRNA, extracellular vesicles, and more. And we are based in Ajdovština, Slovenia. This is close to Italy, close to Austria and Croatia. We are about 2 hours from Venice, for example. So this is on the picture, you can see our production facility. Besides the manufacturing, we also offer solution for downstream processing and solutions for analytical methods development. So why are monoliths ideally suited for purification of large molecules such as extracellular vesicles? So compared to porous particle media that have diffusive mass transport, the mass transport in monoliths is convective. This means that the resolution and capacity are flow independent, meaning that the scale up is really simple. So the also small porous particle media have pores. Pores are usually small, which limits the capacity for large molecules. But monoliths have channels, which means that the surface area of channels is more accessible, offering high capacity for large solutes. Also, there is no dead-end pores, meaning that there is no entrapment of the solutes. In packed bed media, there are usually eddies that cause shear forces, which impacts the yields of shear sensitive products such as extracellular vesicles. But in monoliths, the flow is exclusively laminar, which means there are no shear forces, which means higher yields. And also the resolution is better because there is less diffusion and less turbulent mixing. We offer different products. So most importantly, products in preparative chromatography. CIMmultus columns are offered in scalable formats from 1 milliliter up to 40 liters. They're ready to use, so no packing is needed. They're multi-use columns. We also offer CGMP compliant columns, and the majority of chemistries are stable for cleaning in place with 1 molar sodium hydroxide. We also offer screening on 69 well plates. 69 well plates are optimal for high throughput screening and analysis and design of experiments. And on the analytical part, we offer CIMac analytical columns. CIMac columns are packed in stainless steel. They are high resolution monoliths. They have 100 microliter bed volume, and the analysis using them is offer short analysis time. There is no carryover, and we also offer specialized formats for CIMac pDNA, CIMac AAV, adeno, and so on. We also offer a process analytical technology HPLC platform called PATfix system. PATfix is designed for in-process control and also for product release quality control. It also has preloaded methods, and there are also reference standards available. We also offer services. Process development services. We offer single methods or start to finish procedures, writing SOPs, training, tech transfers to CMOs, scale up of processes, and also on-site support if needed. The process development focuses on developing methods on envelope viruses, such as influenza, or non-envelope viruses, such as AAV, phages, then extracellular vesicles, pDNA, mRNA, and proteins. And we focus on purification processes based on CIM monoliths. The analytical development services team offers development of methods, either single methods or for full process monitoring, for monitoring product quantity, quality, as well as impurity monitoring. The methods include methods for viruses, extracellular vesicles, pDNA, mRNA, impurities, and other biomolecules. And the majority of methods is developed for analytical HPLC, so PATfix system with CIMac monoliths. We also have a department which focuses on custom monoliths development and manufacture. So they developed immobilization protocols for any ligands, either affinity, ion exchange, hydrophobic interaction, reverse phase. They prepare custom monoliths at any scale, and they also do provision of protocols to customers who prefer to do their own immobilization. Okay, so firstly, we will focus on PATfix HPLC system for process monitoring. PATfix is biocompatible HPLC analytical system. It's specifically designed for rapid analysis and in-process control of both upstream and downstream processes. Applications for extracellular vesicles include detection of extracellular vesicle specific antigen using SEC fluorescence and antibody labeling, detection of EVs with a multi-angle light scattering detector, and also anion exchange analytics of EV samples. First, how do we use size exclusion chromatography analytics on PATfix for detection of extracellular vesicles? So PATfix is a multiple detector system. The technology enables simultaneous detection of absorbance, fluorescence, light scattering, emitted scatters from the sample, which provides a lot of information in one run. Fluorescent detector can be used to detect transmembrane proteins, such as tetraspanins, typically enriched in EV preparations, or basically any molecule on the surface of the extracellular vesicle. We can also do detection of engineered EGFP expressing EVs, and also we can use label-free detection using Multiple-detector. So let's go through the size exclusion analytics workflow. Samples, either upstream samples or different in-process downstream samples, are spiked with specific antibodies that are conjugated to fluorescent probe. In the experiments that I will show, we use, for example, FITC, anti-human CD81, CD63, or CD9, but also other antibodies can be used. And the mixture was incubated until exosomes were bound to exosome antigens of interest. And then we did size exclusion separation. So on size exclusion, the SEC column that was used was TSK gel column from Toso Biosciences. The exosomes are too large to enter the pores of SEC media and are excluded in the void volume, which you can see on the chromatogram, while excess antibody is retained on the column and separated from the product peak. The fluorescent labeled product peak, so the first peak that you can see, is then analyzed using PATfix software. In our experiments, we used EGFP labeled engineered exosomes. So for detection of these, we used a simplified workflow. The samples were simply separated on size exclusion, and the product peak was analyzed, as you can see on the chromatogram on the right. So I mentioned that PATfix is used both for upstream and downstream analysis of samples. We will start with the upstream part, and especially why is testing input materials in exosome production important. So both animal and human-derived components that are used in cell culture, such as fetal bovine serum or human platelet lysate, naturally contain extracellular vesicles or similar particles. So these particles can cross-contaminate the desired product. In order to study EVs derived from the cells of interest, multiple producers have come up with exosome-depleted FBSs. In this example, various FBS products were tested for exosome markers using PATfix size exclusion assay. On the chromatograms on the right, you can see only void volume peaks, so only the peaks where EVs elute. You can see both UVs, so UV to 60, UV to 80, fluorescence for CD9, CD63, and CD81 overlaid on one chromatogram for each sample. Also control without the addition was done, and light scattering detection at 90-degree angle for each sample. So regular FBS is known to contain serum-derived exosomes, and was used as a positive control. On the figure labeled A, you can see CD9 positive EVs in FBS. You can also see response on light scattering detector, showing that here we have particles in this sample. Exosome-depleted FBSs that were intended for our exosome production were confirmed to be free of chosen exosome markers. Only the FBS from producer 1 contained traces of CD9 positive vesicles. Okay, so how did we prepare our EV upstreams? In this example, we used an adherent HEC293 cell line expressing green fluorescent protein stacked tetraspanin CD63. Cells were cultivated in DMEM with high glucose with Glutamax and without phenol red, and supplemented with 10% of VBS. At the first passage, cells were split and then grown for 3 more subsequent passages in the presence of different supplements. On the table, we can see cell density data and viability data of these cells in the presence of these different FBSs. Samples were collected from the first passage after 72 hours of culture. So we can see the viability was high, above 90% in all cases. On the right, you can see the effect of different supplements on HEC293 cells, cell growth. You can see all EV depleted supplements reduce cell growth to some extent. And among EV depleted supplements, the best cell growth was observed with the supplement from producer 3, which is colored yellow on this picture. How does cell growth translate to EV production? So during production, CD63 EGFP is integrated into EVs, which allows detection of these vesicles with fluorescent monitor on PATfix system. Conditioned media samples from previous experiments were here analyzed using PATfix HPLC assay. And you can see the chromatogram on the right. So you can see the size exclusion chromatogram of EVs from HEC culture with regular FBS on labeled A, and on B, C, and D, you can see EVs from HEC culture with EV depleted FBSs from different producers. The highest CD63 EGFP response was measured with EVs with EV depleted FBS from producer free, where we also observed the best cell growth compared to other EV depleted supplements. Moving on to the downstream parts of this presentation. So how did we isolate EVs from this upstream? We used anion exchange chromatography capture, and we used a simplified workflow allowing the detection of EV population in the harvest. So we used HEC exosome harvest. We used a CIMmultus quaternary amine ligand for binding of these EVs and for fractionation. And then we used PATfix for detection of EVs in these samples. So how exactly we isolated EVs from this HEC CD63 EGFP harvest? In this case, we use CIMmultus QA column with 4 milliliter column volume. The porosity of this column was 2 micron. And the sample that we used was 100 milliliter of HEC EV harvest. The load was prepared by dilution with a dilution buffer that consisted of HIPS, sorbitol poloxamir, pH 7.5. The loading buffer was 50 millimolar HIPS, 50 millimolar salt, 1% sorbitol, 0.05% poloxamir, pH 7.5. And the elution buffer had the same composition, just 2 molar salt. The elution was done in 15 column volumes, and the cleaning in place was done with 1 molar sodium hydroxide, 2 molar sodium chloride solution. This was done on a preparative chromatography system on Acta Pure, equipped with 2 millimeter UV cell, and we also used a MULS detector. So MULS was used to accurately collect EV peaks on the preparative run, but it can be misleading if we try to interpret the results solely based on the preparative chromatography. The middle pictures shows the chromatogram, complete chromatogram. We can see a flow through, so everything that did not bind to the column. And we can see the elution in the increasing salt gradient. The elution is here zoomed in on the right. You can see the fractions that were collected, labeled from E1 to E4, and also the cleaning in place. The samples from the previous experiment were analyzed using PATfix size exclusion method. So we can see here that the signals from multiple detectors from PATfix are overlaid on 1 chromatogram for each sample. So the samples that were analyzed here were load, so what we applied to the column, the flow through, so the fraction that did not bind to the column, and then the elution profile, where we can see different populations of EVs eluting. So you can see that absorbance was monitored at 2 wavelengths on PATfix system, 260 and 280. Fluorescence was monitored at emission and excitation settings for CD63 EGFP protein, so for fluorescently labeled EVs. And light scattering was monitored at 90-degree angle. You can see the complete chromatogram. and also the zoom in of the target peak, so only void volumes, only where we see the EVs eluting. So we can see that here we have detection of EVs by fluorescence and light scattering, but we can also monitor the impurity composition by UV signals. Moving on to the next slide. How do we analyze the distribution of fluorescent EVs in the elution fractions? So here you can see the elution profile from the previous chromatogram. You can see that fluorescent EVs were fractionated in subpopulation on CIMmultus QA. And then they were detected using PATfix HPLC fluorescence detector. So the peak heights from the PATfix SEC analytics were accounted for the dilution and the volume and were plotted on this bar graph. We can see that that we didn't detect any EVs in flow-through, so all EVs bounced to the column, and they were separated in the salt gradient, peaking in two peaks, so E2 and E4 is where we detected the most EVs. What other data can we extract from size exclusion chromatography? So in this slide, we compared light scattering analysis from PATfix SEC assay compared to total protein, total particle analysis using NTA. So we can see that EGFP labeled exosomes can be tracked using a fluorescence detector on PATfix system. We can see that MALS and fluorescence analytics correlated well with NTA results. And all analytical methods that we used confirmed that the majority of the EVs particles were enriched in fraction E4 followed by fraction E2. But what about when we have to detect native EVs in the sample? So in this experiment, we used a HEC EV native harvest. In this case, EVs were not fluorescently labeled. With EGFP, we had to additionally label them using antibodies conjugated to a fluorescent probe. So here we illustrate native HEC EVs detected using CD63 specific antibodies conjugated to a fluorescent probe. And we compare that to total particle analysis using NTA. Again, on the bar graphs, we can see that the most particles was measured in E4, followed by E2. And on the fluorescence detection, we can see that the most particles were detected both in E2 and E4 fraction. So to sum up, PATfix size exclusion analytics is a powerful analytical tool for screening both raw materials, so upstream samples, extracellular vesicle harvests, as well as downstream samples and in-process fractions. PATfix SEC complements common process development approaches and speeds up the decision-making process. CIMmultus line, so that was used for preparative isolation of exosomes, offers a rapid and shear-free tool for a concentration and fractionation of extracellular vesicles from harvests. At this opportunity, we would like to thank Simon Staubach, that is now Sartorius, and Bernd Giebel from University Hospital Essen for kindly providing a HEC 293 CD63 EGFP cell line that was used in the experiments. With this slide, I would like to conclude my presentation, and I'd like to thank you for your attention.
Okay, thank you very much, Katja, for sharing this presentation with us. You have shown us interesting tools for extracellular vesicles isolation and analytics, and we already have some questions from the audience. Before we proceed, I would again like to invite everyone to submit questions through the webinar platform so they can be answered by our speaker. Questions are not visible to the audience, and if any question is not answered by the end of the webinar, we will address them by email. So the first question, Katja, you mentioned anion exchange analytics. Which analytical columns do you most often use for analytics of EV samples?
So for among CIMac analytical columns, we use CIMac adeno column. CIMac adeno column is specifically developed for monitoring adenovirus, but because of its properties, is also ideal tool for monitoring EV samples from downstream.
Okay. How can you increase the purity of the final EV fractions? This is our second question.
So in this presentation, I only showed a simplified version of isolating extracellular vesicles. But if we want to increase the final product purity, we can use pre-treatment methods. One of the methods is TFF. So before anion exchange chromatography, we would remove low molecular weight impurities using TFF. And the other method is using CIMmultus OH column, so hydrophobic interaction chromatography, for the pre-treatment of the sample, followed by anion exchange chromatography.
Okay, thank you. So another question is, how many times can you reuse monolithic columns that were used in the experiments?
So this really depends on the sample used. So we typically reuse columns multiple times. If we use the column as a capture step, so the first one after upstream, then the column can be reused less times compared to if we use it for polishing. Polishing columns can be reused many times.
Okay. Another one is, what is the dynamic binding capacity observed under the conditions used in this study?
So for this study, we didn't test capacity of this sample, and dynamic binding capacity really differs for each sample that we test.
But can you evaluate how much could be? You didn't study it, but approximately what are the numbers?
So depending on the sample, if we have sample, sorry, let me think about it. So we typically can bind about e to the 11 or e to the 12 of EVs per milliliter of monolith. But this really depends if the sample is pre-treated before, so that other impurities such as proteins that take up the capacity are already removed, or if the sample, or this is in the capture step. In the capture step, the dynamic binding capacity will be lower.
Okay, thank you. Do you have a recommendation for particle to dye ratio for CD63 antibody labeling?
No, sorry, I don't have the recommendation. So we tested this for different samples. We recommend doing tests with different concentrations and then optimizing this for your sample.
Okay. What was the process time for anion exchange capture of EVs that you showed earlier?
So process times, in the small scale, we load up to 16 milliliters per minute. A small scale run that I showed takes about 20 minutes to complete.
Okay, but what about the whole process, not just loading? So from the start of equilibration till the end of elution?
The equilibration, so the column preparation takes about 15 minutes. Each column is first cleaned, then it's regenerated, and then is equilibrated to binding conditions. Then the sample is applied. So in this case, the sample application took less than 10 minutes. And then the column is washed to remove any unbound components on the column, which takes a few minutes. And the whole elution took about 15 minutes, for example. And then we go again on cleaning in place procedure. So the whole cycle takes about 30 minutes.
Okay, thank you, Katja. I think that's all from our questions. We can wait another minute if anyone remembers to ask something else. And then we will end for today. So yes, if anyone have another questions, please submit them through this webinar platform so they can be answered live, not just via email.
So for more specific questions, we can, if you contact help via separations.com, we can help you answer these questions better.
Okay, so I think this is all for the time we have today. And yes, if there are any unanswered questions, please send them to help@bioseparations.com so we can address them by email. I would like to thank again everyone for joining this webinar and thank Katja Vrabec for sharing EV experience with us. I wish you all a great rest of the day.
Thank you, Masha.
Thank you, Katja.